This is the first in a series of lectures on metabolism of carbohydrates. We will start by examining metabolism of glucose or glycolysis. Metabolism of carbohydrates in general involves digestion of complex carbohydrates to their constituent mono, dry, trisaccharides, and maybe some oligosaccharides, and then they are further hydrolyzed to monosaccharides, which are then metabolized. By far, the most abundant monosaccharide is glucose, and the most abundant polysaccharides are polymers of glucose. So, polymers of glucose are starch or glycogen, and they are hydrolyzed either by salivatory alpha amylase, which is minor amount, or most of them are hydrolyzed by pancreatic alpha amylase to di and trisaccharides and some limit dextrin. Alpha amylase hydrolyzes or cleaves alpha 1,4-glycosidic bonds. That results in formation of disaccharide maltose and trisaccharide maltotriose. Alpha amylase cannot cleave alpha 1,6-glycosidic bond. So branching points of starch or glycogen remain unhydrolyzed, and at each branching point there are several glucose residues that remain. That's called limit dextrin. So those are products of digestion of starch or glycogen by alpha amylase. Maltose, maltotriose, and limit dextrin. And then in turn, maltose is hydrolyzed by enzyme maltase, maltotriose by alpha glucosidase, and limit dextrin by alpha dextrinase. And all of them are hydrolyzed to glucose. Glucose is then further metabolized in sequence of reactions into 2 molecules of pyruvate and 2 molecules of ATP. And that's the process known as glycolysis. Glycolytic pathway involves 3 stages, and those are 3 stages, not 3 steps. Each of those stages consists of multiple steps, of several steps. In the first stage of glycolytic pathway, glucose is converted into fructose 1,6-bisphosphate. In the second stage, fructose 1,6-bisphosphate is cleaved into 2 3-carbon fragments, and in the final third stage, those 2 3-carbon fragments are oxidized and ATP is generated. In the first stage of glycolysis, glucose is converted into fructose 1,6-bisphosphate. So after polysaccharides are hydrolyzed to monosaccharides, which happens in the small intestine, they are absorbed into the bloodstream. Monosaccharides, other than glucose, are first converted to glucose and then metabolized as glucose. Glucose is then transported into the cell by transport proteins, and then in the first step it's phosphorylated by ATP. So glucose 6-phosphate itself is not a substrate for glucose transporters, and so cannot pass through the membrane out of the cell. It's also less stable than glucose, and then it means that it's more easily metabolized. So phosphorylation of glucose into glucose 6-phosphate, which is catalyzed by enzyme hexokinase, commits glucose to metabolism. So this is committed metabolic step, but it does not commit glucose to particular metabolic pathway. It only commits glucose to being metabolized. Glucose 6-phosphate can undergo a number of different metabolic pathways. This enzyme hexokinase catalyzes phosphorylation. In general, any enzyme that is named kinase catalyzes phosphorylation. Fructose 1,6-bisphosphate is substrate only for glycolytic pathway. So formation of fructose 1,6-bisphosphate is going to be the committed step of glycolytic pathway. Now, before fructose 1,6-bisphosphate can be catalyzed, glucose 6-phosphate first has to isomerize in a reversible step, so obviously catalyzed by Michaelis-Menten enzymes So enzymes that are not controlled. Enzyme is phosphoglucose isomerase. Glucose 6-phosphate isomerizes into fructose 6-phosphate. Isomerization occurs through open chain form. And then fructose 6-phosphate is phosphorylated by the enzyme phosphofructokinase or PFK into fructose 1,6-bisphosphate. So this step, catalyzed by phosphofructokinase or PFK enzyme, is the committed step of glycolysis. In the second stage of glycolysis, fructose 1,6-bisphosphate is cleaved into 2 3-carbon fragments. The reaction is a retroaldol reaction. So this is where knowledge of organic chemistry comes in really handy, and hopefully you remember your organic chemistry course. And that means hopefully you remember aldol reaction. If you do, then first you know what aldol reaction is, and you know that an aldol reaction is reversible reaction. So reverse of aldol reaction is retroaldol reaction. Just to remind you, an aldol reaction is a condensation reaction, or actually this condensation would be aldol condensation. Aldol reaction is a reaction where aldol is produced. Aldol means aldehyde alcohol, so aldol. Condensation is when that alcohol undergoes elimination and we obtain alpha-beta unsaturated ketone. So that's just to remind you, that would be more common reaction. So aldol condensation is more common, but first step of aldol condensation is an aldol reaction. In an aldol reaction, an aldehyde is deprotonated in position alpha to carbonyl group, and that resulting nucleophile adds to another molecule of the same aldehyde to form new carbon-carbon bond, to another molecule of the same aldehyde that has not been deprotonated. Only a few of the molecules of the aldehyde are deprotonated. The result is formation of new carbon-carbon bond and the intermediate alkoxide, which is protonated to give the product the corresponding aldol. Each of those steps is reversible, and so reaction can occur in reverse. So starting with an aldol, in retroaldol reaction, aldol is deprotonated, hydroxyl group of aldol is deprotonated, and then carbon-oxygen double bond is formed or reformed. If we look at original starting compounds, carbon-oxygen double bond is formed to give carbonyl compound, and that's accompanied by simultaneous breaking of the carbon-carbon bond. The result is formation of 2 fragments, an aldehyde and an inlet anion. Inlet anion is protonated to give the other molecule of the aldehyde. We can apply that to cleavage of fructose 1,6-bisphosphate. We should identify the bond that is going to be broken, and that's starting with carbonyl group, that's 2,3 bond of the aldol. Or in this case, because it's fructose, it's actually a ketol. It's ketone aldehyde. So it's 2,3 bond shown here in red of the ketol. In this case, instead of a strong base, reaction is catalyzed by the enzyme aldolase. Upon cleavage of the indicated bond, 2 3-carbon fragments are formed. They are dihydroxyacetone phosphate, or DHAP, and GAP, glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is substrate for glycolysis and enters glycolysis as such. Dihydroxyacetone phosphate is not substrate for glycolysis and has to be isomerized. So carbonyl groups and hydroxyl group basically exchange place in isomerization reaction. Reaction is catalyzed by triose phosphate isomerase enzyme, where dihydroxyacetone phosphate is isomerized into glyceraldehyde phosphate or GAP. Of course, an enzyme cannot shift position of the equilibrium. It can only speed up the reaction, isomerization of the two. Equilibrium position is to the left as dihydroxyacetone phosphate. So at equilibrium, mixture is composed of 96% of dihydroxyacetone phosphate and only 4% of glyceraldehyde 3-phosphate or GAP. But that GAP enters glycolysis and is constantly being removed from the equilibrium. And according to Lesch-Tolier's principle, if it's being removed from the equilibrium, then it's constantly going to be replenished by isomerization of more GAP. And so that isomerization constantly happens, GAP is being removed, goes into glycolysis, until eventually all of the dihydroxyacetone phosphate enters glycolysis as GAP. And here you can see a mechanism of retroaldol reaction of fructose 1,6-bisphosphate, how it would occur if it were to be carried out in a flask in the presence of a strong base. Products would have been the same, dihydroxyacetone phosphate and GAP. Now, had the retroaldol cleavage occurred in glucose, then products would have been 2-carbon and 4-carbon fragments, and that would have required 2 different metabolic pathways for further metabolism, for further processing, and it would have been very inefficient. So that's the reason behind whole first stage of conversion of glucose, first stage of glycolysis, of conversion of glucose into fructose 1,6-bisphosphate, so that fructose 1,6-bisphosphate can be cleaved into 2 3-carbon fragments, rather than starting with glucose and ending up with 2 different fragments, 2-carbon and 4-carbon fragments, which would then require 2 different metabolic pathways. These first 2 stages of glycolysis generate no energy. In fact, 2 molecules of ATP have been consumed to produce fructose 1,6-bisphosphate. So these 2 stages actually require energy input. So they consume energy. So initial digestion of food actually requires energy. And that's behind so-called refeeding syndrome. That occurs if humans or animals have been starving. So if they have been really starving and then are given a large amount of food, that could actually harm them. They could even die because they are very low on energy, and then those remaining energy reserves may be used up, completely consumed in these first stages of metabolism, and actually energy for vital living functions may not be there. Formation of GAP and dihydroxyacetone phosphate in liver and skeletal muscles may deplete completely intracellular ATP and 2,3-diphosphoglycerate in red blood cells. And so result is cellular dysfunction and inadequate oxygen delivery to body's organs. And then insulin is released in response to increased levels of glucose, and that results in increased glycogen, fat, and protein synthesis. And so that requires phosphate, magnesium, potassium, which are already depleted, and any remaining reserves are rapidly used up. So here I have an example of a kitten that's about, my estimate is 6-week-old kitten that apparently was abandoned by her mother because she was sick. And when I found her, she was actually skin and bones. She was incredibly tiny and just basically a skin and bones. Here she is after having a bath, so one can more easily see how tiny she is and how skinny she is. And she was given a small meal. Even though she was starving, she was given only a small meal, only about a quarter of a small can of cat food. But then she was given repeated small meals. Every few hours, approximately every 4 hours, she was given a small meal until she built up her energy reserves. And then she could be fed normally and could be allowed to free feed. But initially she definitely should not be allowed to free feed. If one were to feed her milk or KMR, kitten milk replacement, then this size of kitten would need about 8 milliliters per meal, 8 milliliters of milk or kitten milk replacement per meal. And so every 3 to 4 hours. And very quickly, within about 2 weeks, she already kind of plumped up and was out of danger, and by about 6 months, she was actually quite good, healthy kitten. So if humans or animals who were starving, should be given small amounts of food, small meals, and if possible, they should be accompanied by large amounts of electrolytes and B vitamins. B vitamins are coenzymes of the digestive enzymes. In the final third stage of glycolysis, 3 carbon fragments are oxidized and ATP is generated. So this is when some energy is generated. So GAP is oxidized and phosphorylated in the presence of nicotine amide adenine dinucleotide and inorganic phosphate. Enzyme is glyceraldehyde 3-phosphate dehydrogenase. Product is 1,3-bisphosphoglycerate on or 1,3-BPG and NADH and protons. This reaction involves 2 steps. First step is oxidation of glyceraldehyde 3-phosphate to 3-phosphoglycerate, and this is favorable reaction. It has delta G of minus 50 kilojoules per mole, so extremely favorable reaction. Second step actually is formation of mixed anhydride, and that's an endothermic step. That's not a favorable step. Formation of mixed anhydride is 1,3-bisphosphoglycerate. It's anhydride of carboxylic and phosphoric acids. If you look at reaction diagram, you can see first step that we have seen on the previous slide, going from GAP to 3-phosphoglycerate, extremely exergonic step, so highly favorable step. And then in the second step, 3-phosphoglycerate is converted in an endergonic reaction to 1,3-bisphosphoglycerate, and also there is kinetic block for that reaction. Enzyme overcomes that kinetic block and speeds up the reaction by converting 3-phosphoglycerate into corresponding thioester. This is what thioesters are. Formula of generic thioester is shown on the right. So thioesters have a better leaving group. So it's SASR leaving group. So that's a better leaving group. They are less stable compared to ordinary esters, and so in terms of energy content, their energy content is higher. So that means that activation energy is lower, and as a result, reaction is faster. And so it's thioester that is eventually converted directly to 1,3-bisphosphoglycerate. So reaction proceeds in such a way that 3-phosphoglycerate, after oxidation, is first converted to thioester, and then thioester is phosphorylated to 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate is one of the compounds that has high phosphoryl transfer potential. Compounds with high phosphoryl transfer potential are compounds that have phosphoryl transfer potential greater than that of ATP. So they are products of various metabolic pathways, and then they transfer phosphate group to ADP, adenosine diphosphate, to generate ATP. So a number of metabolic pathways generate compounds with high phosphoryl transfer potentials, which then in turn are converted to ATP. In this case, 1,3-bisphosphoglycerate is the compound that transfers phosphate group to ADP. Enzyme is phosphoglycerate kinase, and product is ATP. Byproduct is 3-phosphoglycerate. Phosphoglycerate is then converted to pyruvate. It is converted to pyruvate in a series of steps. First, phosphoglycerate mutase exchanges phosphate position, exchanges group where the phosphate is attached. It moves phosphate from position 3 to position 2 of glycerate. So converts 3-phosphoglycerate to 2-phosphoglycerate, and then enolase generates an enol compound of it, phosphoenol pyruvate. Phosphoenol pyruvate is another compound with high phosphoryl transfer potential, because as an enol, it is unstable and has high tendency to donate phosphate group. Enzyme is pyruvate kinase, and so phosphoenol pyruvate transfers phosphate group to ADP to generate another molecule of ATP. So both phosphate groups from starting 1,3-bisphosphoglycerate have been now transferred to ADP to generate 2 molecules of ATP. product is pyruvate. So 3-carbon fragment has generated 2 molecules of ATP. Keep in mind that glucose generates 2 3-carbon fragments, which means that overall 4 molecules of ATP were generated. But remember, 2 molecules of ATP were initially needed to synthesize fructose 1,6-bisphosphate. So net result is that overall glycolysis, net glycolysis generates 2 molecules of ATP. So it's an anaerobic process and it actually generates relatively small amount of energy, only 2 molecules of ATP. And also 2 molecules of pyruvate are generated. For glycolysis to continue, NAD has to be regenerated. Now it is in reduced form, NADH, and so as a cofactor, catalytic cofactor, it has to be regenerated. There are 3 ways to regenerate NAD, or 3 pathways. 2 of them are anaerobic, and they are called fermentations. In fermentations, organic compounds are both acceptors and donors of electrons. And third is aerobic, and involves oxidation of acetyl coenzyme A with oxygen that is acceptor of electrons. So in one possible fermentation pathway, which is a relatively minor pathway that occurs in yeasts and some other microorganisms, ethanol is formed. So pyruvate, upon action of enzyme pyruvate decarboxylase, releases carbon dioxide, is decarboxylated, so it loses a molecule of carbon dioxide and gives a molecule of acetaldehyde. And now NADH, which was generated in the course of glycolysis, and proton are used up in hydrogenation reaction to hydrogenate acetaldehyde to ethanol. And ethanol is the final product of fermentation, of this fermentation. Enzyme is alcohol dehydrogenase. That's a relatively rare pathway, but it's used, as you know, in brewing of alcoholic beverages. So relatively few organisms pursue that or follow that pathway. Much more common fermentation pathway is formation of lactate. So lactate is formed by a variety of microorganisms in lactic acid fermentation, and also it takes place in higher organisms, including humans, under anaerobic conditions. In that case, pyruvate, upon action of enzyme lactate dehydrogenase and with NADH and proton, is hydrogenated into lactate. Now, lactate is metabolic dead end. In humans, it is exported into the blood and carried to the liver for gluconeogenesis and then eventually back for production of glucose. Much more energy is obtained under aerobic conditions, and that's so-called citric acid cycle, which we'll be covering a little bit later in this course. So in this pathway, glucose is oxidized all the way to carbon dioxide and water. The entry point into the citric acid cycle is formation of acetyl coenzyme A from pyruvate, and we will be covering that a little bit later in this course in great detail. And that occurs inside the mitochondria. So pyruvate with NAD and coenzyme A is converted into acetyl coenzyme A. A molecule of carbon dioxide is released, and NADH is regenerated in the electron transport chain in mitochondria. This is the most efficient way and generates by far the most energy. In fact, the most energy is generated this way, and only minor amount of energy has been generated in anaerobic pathway in glycolytic pathway.